Okay, welcome all. Thank you for being here for the Northwest Regional Invasive Species and Climate Change, or RISC, Management Network's fall webinar, Connecting the Dots from Forest and Estuary Management to Climate Resilient Salmon. I'm Eva Kohlberg, Northwest RISC Coordinator, and I'll be moderating today's webinar. I'm also joined here by our two speakers, Michele Buonanduci, who will be talking about how land-based restoration actions can contribute to the resilience of salmon populations under climate change, and Chad Phillips, who'll be talking about the Washington Department of Agriculture's Spartina eradication program. And you'll hear from both of them in just a moment. But I also wanted to say we're joined by Darcy Gould, who'll be fielding any technical questions you have. If you're having tech issues, go ahead and put that in the chat box, and Darcy can deal with that. But I also wanted to give you a little bit of background of what Northwest RISC is. We're dedicated to connecting practitioners and researchers across the Northwest in order to address challenges that are at the nexus of climate change and invasive species, and really from a management and action-based, science-informed action perspective. For today's webinar, we will be recording and we'll be posting this webinar onto the Northwest RISC website, as well as our YouTube page, so you can come back and get that information if you forgot to take notes or wanted to come back to some part of it. And if you're joining us live today, there are a couple different ways to interact. You can ask questions in the Q&A feature, and we'll take some time after each talk in order to answer those questions. And you also should have the ability to react via Zoom, and so there should be a little feature in your toolbar that should allow you to show some love. I'm seeing hearts, so it looks like that's working. If you don't want to see other people's reactions, you can also use the little caret next to that reactions button on your toolbar to just hide everyone else's reactions to reduce distractions. I did also want to clarify that we have had another webinar scheduled for tomorrow, but we've rescheduled that. So our Northwest Weed Biocontrol and Changing Environments webinar will now be January 27th from 12:30 to 1:30 p.m. Pacific time. And if you already were registered, no worries, that Zoom link is going to transfer and update. But if you want to register for that, you now have more time to do so. And so we'll put the link in the chat in a moment. And finally, that means that our next RISC event will actually be co-hosted with the other RISC networks. This will be our International Invasive Species and Climate Change Conference that will be held the second week of December. So look forward to that. But for now, let's return to the topic at hand. I'm going to pass things over to Michele.
Great. Thank you so much, Eva, for the introduction and the invitation to be here today. Let me get my screen sharing started. Okay. Hopefully you should be seeing my title slide. So today I'm going to be talking about Willapa Basin salmon responses to land management in the context of climate change. Before I dive in, I want to acknowledge my collaborators on this work. This was a collaborative effort between myself as well as some other folks at the Nature Conservancy in Washington, as well as collaborators from WDFW and NOAA. So to orient everyone to the study I'll be talking about today, here we're looking at the Willapa Basin, which is shown here on the left in this blue color. And this is in the very southwest corner of Washington. This is a very heavily forested area that has been managed pretty intensively for timber production over the last century. And so presently, very few pockets of old growth forest remain in this area. The yellow patches you see in this map actually indicate the few remaining old growth forest areas in the Willapa Basin. So you can see there's not a whole lot there. Now, back in the year 2000, TNC in Washington bought the entire Ellsworth Creek watershed, which is down here towards the southern end of Willapa Bay. And by purchasing the entire watershed, TNC was able to preserve some of these really critical remaining pockets of old growth forest, while also having the opportunity to experiment with ways to restore large portions of the watershed that had been heavily logged for decades. So experimenting with ways to take young, dense, simple structure forest stands that look something like this, and moving them towards something that resembles some of the really iconic remaining patches of old growth forest that we have out at Ellsworth. So for TNC, this preserve is what they like to call a living laboratory, where they are both implementing restoration projects as well as monitoring and testing for restoration success. And one of the ways TNC is interested in measuring restoration success in this watershed is through understanding what all of the restoration work out here has meant and might continue to mean for salmon. Because Ellsworth Creek, which you can see in this aerial photo here, is used by several species of Pacific salmon for spawning and reproduction. Now, Pacific salmon, as you are likely aware, are broadly in need of restoration action. This is due to a wide range of factors, including things like habitat loss and degradation, barriers to fish passage, detrimental fisheries practices, and increasingly also climate change. As you may also know, the salmon life cycle is really complex. So salmon require a mosaic of habitats ranging from streams to estuaries to oceans and back again to complete their life cycle. So we know that what we do on land through our habitat restoration and land management can potentially benefit salmon by improving salmon's spawning habitat and juvenile survival. But we also know that climate change is going to put pressure on salmon really across the board at each stage of their life cycle. So we can't evaluate how our land management might be affecting salmon without also taking climate change into account. So with that context, in light of all of the restoration work out at Ellsworth over the last 25 years and more broadly across Willapa Bay, the research questions we wanted to address in this study were first, how do concurrent land-based restoration strategies affect salmon populations within the context of climate change? And then more specifically, what is the potential for forest management to increase the resilience of salmon to climate change? And we addressed both of these questions using a life cycle modeling approach for chum salmon in the Willapa Basin. We partnered with the Washington Department of Fish and Wildlife for this work. They monitor the salmon populations in Ellsworth Creek, as well as within two other watersheds in the Willapa Basin. So first there's Ellsworth, where, as I just described, land management over the past 25 years has been focused on forest restoration for a variety of objectives. And then there are these other two watersheds. These watersheds are owned primarily by private industrial timber companies, and so understandably, the management in these watersheds has continued to be focused on timber production. So in our modeling, we were able to harness the information from across these different contexts to try to understand how salmon are responding to different types of forest management at the watershed scale. At the heart of our modeling approach, we have a life cycle model for salmon. And this is an age-structured statistical model that accounts for the density-dependent relationship between spawner abundance and subsequent recruit abundance within each population. The most critical input into this model is salmon spawner abundance over time, as well as age composition for spawning adults. And then other inputs can include really any number of habitat conditions that we expect might modify recruit abundance or the relationship between spawners and recruits over time, either through changes in habitat quality or quantity. So in this project, we chose to account for forest structure within those three watersheds that I just showed. We also accounted for estuary conditions within Willapa Bay, as well as hatchery releases in the basin. And ocean conditions off the Washington coast. And I'll describe all of these various inputs in a little bit more detail in a moment. After fitting our model to these different data sources, we are able to identify whether and how the populations are responding to these different factors. And finally, we're able to use what the model learns about these different relationships to project salmon population dynamics under different future climate and management scenarios. So getting into the data and starting with our chum salmon monitoring data, we're really fortunate that WDFW has been monitoring the chum populations in Willapa Bay for decades now. So this figure here shows a time series of chum spawner abundance in those 3 watersheds since 1984. And again, this serves as one of the primary inputs into our salmon population model. Next, to account for forest structure within our 3 watersheds over time, we're using an old growth structural index that's available for our study area. And this is an index that reflects the abundance of large live trees, snags, and down wood, as well as the diversity of tree sizes. And these are all factors that can affect salmon survival and productivity via effects on runoff, flow, sedimentation, shading, and woody inputs to streams. So showing what these data look like, the 3 watersheds are shown in the 3 colors here. I'll just point out that we have Ellsworth in the black line, and you can see that after the year 2000, when TNC acquired the preserve, the watershed, forest structural complexity has been steadily increasing over time. Compare that to the other 2 watersheds where there was a lot of clear-cut harvesting in the mid-2000s, and you can see the steep drops in forest structural complexity during those years. Next, moving to estuary conditions. A really notable change in estuarine habitat in Willapa Bay during our study period was the spread of Spartina, which is an invasive species of cordgrass, and which expanded really rapidly through the estuary in the early part of our study period, peaking in the early 2000s. We suspected that Spartina could have affected our study populations of chum by altering sedimentation patterns in the estuary and potentially by competing with or altering native habitats, particularly native eelgrass habitats that juvenile chum really rely on for foraging and predator avoidance during a particularly vulnerable developmental period in their life cycle. Fortunately, some really concentrated and cooperative efforts to control Spartina in the early 2000s were largely successful. Chad is going to tell you about that in a lot more detail after I finish up my talk. But you can see that Spartina acreage in the estuary dropped sharply in the early 2000s and is currently near 0 and expected to remain near 0 with continued control and eradication efforts. Next, thinking about hatchery releases in the Willapa Basin, there are 3 hatcheries that release hatchery-reared juvenile chum in the basin. Hatchery origin chum have the potential to compete with wild populations for resources and therefore could potentially negatively affect natural recruit abundance. On the other hand, hatchery releases also have the potential to inflate natural origin spawner counts if rates of straying are high. So to account for and test for a potential effect of hatchery releases on natural recruitment, we included these basin-wide hatchery releases reported by WDFW in our modeling. Finally, moving to the ocean, we accounted for sea surface temperature off the Washington coast. Sea surface temperature is expected to regulate salmon growth and survival during the marine stages of their life cycle. And as you likely know, sea surface temperature is expected to rise through the end of the century. We also accounted for coastal upwelling off the Washington coast. So during upwelling, winds push surface waters away from the shore, and they are replaced by cold, nutrient-rich water that wells up from below. And this process is a strong driver of ecosystem productivity in the nearshore environment, and so is expected to regulate prey availability for salmon. So as you can see, and as you've likely gathered by now, this is a data-driven model. So here's just a summary of our various data sources and inputs into our model. And I'm not going to repeat these or go through them again. Really, I just want to highlight on this slide that the study would not have been possible without all of the research and long-term monitoring being led by state and federal agencies. So moving on to some of our results, again, the first step of looking into our results is identifying how our study populations are responding to all of these different factors. So moving through some of our findings, starting on the left with sea surface temperature, we found that sea surface temperature had a negative effect on the productivity of our study populations. And coastal upwelling had a positive effect. And both of these findings align really well with what we expected to find for these two factors based on what's already out there in the literature. We found essentially no effect of hatchery releases, either positive or negative, on our study populations. This could be in part because hatchery releases are much lower than our estimates of natural production in the basin. So they're actually not super likely to either compete heavily or inflate our counts of the natural populations. Then, looking at estuary conditions, we found that the spread of Spartina through the estuary had a negative effect on the carrying capacity of our study populations. The other way you can think of this is that the really successful Spartina eradication efforts in the estuary had a positive effect on the productivity of our study populations. And finally, we found that watershed scale for structural complexity had a positive effect on the productivity of these salmon populations. Again, the other way you can think of this is that the intensive clear-cut harvesting that happened in two of those watersheds had a negative effect on the productivity of those populations. Okay, so again, what's great about our model is that we can use what we have learned about the observed relationships between chum abundance and these various habitat conditions to make some future projections. And to do that, we need to make some projections for what these different habitat conditions might look like in the future. Okay, so first, starting with sea surface temperature, we used 4 different global climate model derived projections for sea surface temperature off the Washington coast. These projections serve as the 4 climate scenarios in our study, and they differ in the extent to which sea surface temperature is projected to increase through the end of the century, as well as the sort of interannual variability of sea surface temperature over those years. For coastal upwelling, we used just one future projection for upwelling, which is not expected to change substantially off the Washington coast through the end of the century. So you can see that here. As far as the Willapa specific habitat conditions, we assumed that Spartina acreage in the estuary will stay near zero with continued control and eradication efforts through the end of the century, and that hatchery releases will just stay near current basin-wide release levels. For forest structure, we developed 2 future scenarios just for the Ellsworth Creek watershed. So first we have the ecological forest management scenario, which is shown in the sort of light green line here. in which we're just conservatively assuming that forest structural complexity stays at the maximum observed level. Obviously, we expect it will continue to increase as forests continue to develop and change, but we don't really have a defensible way to project what that future trend might look like. Now, this scenario could include some selective harvest or thinning for a variety of objectives, but the idea is that overall forest structural complexity remains relatively high. Compare that to the industrial harvest scenario, which is shown in this gray line here, which assumes that starting in 2035, TNC decides to do a bunch of clear-cut harvesting out at Ellsworth, causing forest structural complexity to drop sharply and then subsequently recover. So to be clear, TNC does not plan to intensively harvest the Ellsworth watershed for timber, but we constructed these two scenarios as sort of bookends to try to quantify the extent to which choosing ecological forest management practices over industrial timber harvesting might increase the resilience of these populations to future climate change. Okay, so getting into some of our findings from our projections. One of the key metrics that we use to interpret our population projections is the quasi-extinction probability for Ellsworth chum under each future scenario. So this is essentially the probability of local population collapse in the Ellsworth watershed through the end of the century. So that's going to be on the y-axis here, and higher values will indicate a higher probability of the population going locally extinct. We have on the x-axis here the climate models for sea surface temperature. Again, these represent our 4 climate scenarios. And then the 2 colors are going to represent our 2 forest management scenarios. So first, when I fill in the estimates from the industrial harvest scenario, the first thing that you'll see is that there are pretty large differences across the climate scenarios, with quasi-extinction probabilities exceeding 10% in the most extreme warming scenario that we considered. Just for reference, from a population viability standpoint, NOAA guidance suggests that quasi-extinction probabilities below 5% over long timeframes such as these are considered acceptable. So you can see we have 2 climate scenarios that exceed that threshold and 2 that are below it. Next, when I fill in the estimates from the ecological forest management scenario, what you can see here is the buffering effect that choosing this management option offers in terms of offsetting some of the negative effects of climate change. And you'll notice that in this climate scenario in particular, that buffering effect makes the difference between an acceptable and an unacceptable risk of local population collapse. Okay, so wrapping this all up with some key takeaway points. First, I think this study really illustrates that land-based restoration strategies do have the potential to increase the resilience of salmon populations to climate change. So specifically, one of the things we demonstrated and focused on is that the importance of forest management strategies that maintain and or increase forest structural complexity. Also, continued estuarine restoration and Spartina control efforts in Willapa Bay will be really critical for these populations, as well as in-stream habitat restoration, which is an important complement to these other efforts and wasn't something that we focused in on in this study. Next, I think this study highlights that forest structure can affect salmon at watershed scales. This is via effects on, again, runoff flow, sedimentation, shading, woody inputs to streams, and really highlights the importance of watershed scale forest management that considers the importance of upland forests in addition to just the riparian zone. Finally, this study does add to the growing body of evidence that climate change has and will continue to put pressure on salmon populations. For that reason, it's important not only to address climate change, but also to invest in land-based restoration strategies in the near term that can increase the resilience of salmon to continued stressors from climate change. With that, I think I have a little bit of time for questions. I've put my email on here in case you want to reach out after the webinar. And then the QR code here will take you to the paper where we publish the results of this study. It's open access, so you should be able to read it, download it in full for free.
Thank you so much, Michelle. That was a great talk. And we do have a few questions. I'll just remind folks to put their questions into that Q&A box if you have others. But we'll go ahead with that first question, actually. So we have a question about what life cycle model is being used, and I think this is specifically for the chum. But then also a question of whether that was considering other invasive species, and I'll just maybe expand on that, of maybe why you were choosing, kind of why you didn't choose other potential factors.
Mm-hmm. Yeah, great questions. So the life cycle model we're using is an integrated population model developed for Pacific salmon and has been developed by Eric Buell and Mark Sherrill and others. There's a great NOAA technical report that came out in 2018 that talks about the development of that model. So I can maybe drop a link to that in the chat after I'm done answering questions. But yeah, it's a statistical model specifically developed to account for the life cycle and age structure of different species of Pacific salmon. And it's a really great tool for exploring how salmon might be responding to not only habitat conditions, but different management strategies. As far as consideration of other invasives, I think we focused on Spartina mainly because it was one that in our early conversations with collaborators just kept coming up as like, oh, this was a really big thing that happened in the Willapa estuary that you should be accounting for in your model. I know a little less about green crab, so I can't speak directly to that. It was one that came up in some conversations early on. My understanding, and please correct me if I'm wrong here, is that green crab has become a problem in the estuary only in more recent years. And I believe that might have been part of the reason why we didn't account for it, because we just didn't have as much, recent data to be able to tease out some of those relationships. Yeah.
Thanks. And maybe we'll go with, this is maybe a generous question to answer in the next couple minutes. But why did you select the specific climate models that you did?
Yeah, yes, that is a really good question. I guess the short answer is we chose them to represent a range of different future conditions, specifically spanning the axes of like less to more, lower to greater future warming, as well as lower to greater changes in precipitation patterns. So we didn't end up accounting for, we were originally going to account for climate change effects in the terrestrial environment as well, which is why we're thinking about precipitation patterns when we were selecting climate models. I won't get into the reasons why we weren't able to do that, but it didn't end up being feasible. But we just stuck with those 4 climate models because they did represent a good range, not only of the sort of magnitude of future changes in sea surface temperature, but also sort of the interannual variation, because the peaks and lows can be important as well as the overall mean trend. Yeah.
Well, thank you so much, Michelle. And if anyone has other questions for you, those can still be put into the Q&A box, just specify that they're for Michelle. But I'll pass things over to Chad to delve into the Spartina side of things. Thank you so much.
Okay. Are you hearing me? And are you able to see my presentation?
Yes.
Okay. Well, that was really a great presentation. I've read your paper and thank you. It makes a lot more sense to me now. So, hello, everyone. Thank you for the opportunity to talk to you about the Washington State Spartina eradication effort. We're very proud of what the program has accomplished. Today, I want to speak to the Spartina threat, some program history, accomplishments to date, and some new challenges. Washington State's invasive Spartina species are salt-tolerant grasses that grow in the intertidal zone along beaches, in estuaries, and on mudflats. Michele just described some exciting findings, including a correlation between invasive Spartina removal and chum salmon increases in Willapa Bay. Here's a photo of that infestation from 2001. A solid Spartina meadow, or a sward, has taken over an intertidal mudflat. No salmon is making its way through that dense growth. Spartina alterniflora was introduced to the Willapa Bay in the 1890s, associated with imported East Coast oysters, eventually expanding to over 8,000 acres and infesting 3 counties. It caused significant impacts to shellfish, including oysters, clams, crabs, migratory waterfowl, and negatively impacted the salmon fishery. Currently, the infestation is comprised of a few hundred plants intermixed with native species scattered throughout the bay. Here's a single Spartina alterniflora seed head containing 10 to 15 spikelets. Each spikelet can have multiple flowers, but typically produces a single viable seed. A Spartina plant can produce hundreds of seed heads, so a relatively typical 4-foot diameter plant can easily produce over 1,500 seeds. Try to imagine how many seeds were produced at the height of the infestation by the meadow in the last image. Spartina spreads. It doesn't respect boundaries. Spartina spreads by both seed production and clonally. Clonal spread occurs when Spartina sends out rhizomes, a form of underground stem that enable the plants to expand in yearly concentric circles until the circles meet, forming a solid meadow. Spartina invasion rates, taking both clonal and seed spread into account, have been estimated at 12 to 17% per year. Here, Spartina clones are spreading and beginning to coalesce in the Willapa mudflats in the early 2000s. Spartina species act as foundation species, dominating the habitats in which they live. While foundation species are beneficial where they are native, they can be especially impactful to invaded systems, modifying valuable habitats and impacting ecosystem services. In this image, you can see a solid Spartina meadow backed by the tree line, clones spreading and joining in the middle, and outliers in front, starting from seed and invading the outer mudflats. Washington's Spartina eradication program protects habitat for native species. Left unchecked, Spartina outcompetes native vegetation and converts mudflats and estuaries into monotypic Spartina meadows. As a result, important habitat for salmon, forage fish, and vertebrates, shorebirds, and waterfowl are lost. The threat of flooding is increased, and the state's shellfish industry is severely impacted. In addition to the Spartina alterniflora found in Washington's coastal estuaries, there are 3 other invasive Spartina species found in Washington state. Spartina densiflora, discovered in Grays Harbor and Island counties in 2001. Early detection annual survey and removal efforts have limited Spartina densiflora impact to Washington's native species. The Island County infestation has been eradicated. The Grays Harbor infestation persists at a few sites, though at low levels. Spartina patens discovered along Hood Canal in the 1990s. Annual survey and treatment efforts have limited impacts and spread, though eradication of this cryptic grass remains elusive. And Spartina anglica, intentionally introduced from the United Kingdom in the 1960s to stabilize dikes and provide forage for cattle. By 1997, the Puget Sound infestation had expanded to more than 1,000 acres and infested estuaries in 10 counties. Currently, the Spartina anglica infestation is the largest in Washington, with an estimated 9 solid acres remaining. These acres are comprised of over 30,000 individual plants or clones intermixed with native species spread across 8 counties. In addition to the negative impacts previously seen from large Spartina infestations, Spartina anglica is emerging as a threat to expensive salmon restoration efforts, where it's shown the ability to quickly invade recently restored habitat following dike removal projects. These invasions threaten to mechanically limit access to the juvenile salmon that need these restored estuaries to prepare for their transition to the saltwater phase of their lives. Spartina threatens Washington's 3,000-mile intertidal shoreline and numerous valuable estuaries. The program cooperators survey thousands of acres of at-risk estuaries every year. Very few of these beaches, estuaries, and mudflats are idyllic and easily accessed. Most are rough, covered in intertidal ditches, muddy, and strewn with logs. Access typically requires a long hike, paddle, or specialized boats and amphibious vehicles. On this slide, you can see an AmeriCorps, Nature Conservancy, Snohomish County, and WSDA staff surveying an estuary in the North Puget Sound, looking for Spartina anglica intermixed with native sedge. Here, proud AmeriCorps crew members pose with a large Spartina anglica plant they found in native sedge and rush. Many plants are shorter than the surrounding vegetation and not nearly as photogenic as this one. In fact, here's a single stem Spartina anglica plant, barely visible after pulling back the much taller native sedges that were hiding it from view. If missed over time, these small plants can completely displace our native species. Plants like this are why we sometimes describe the job as looking for grass in grass. Or a small Spartina anglica plant found in logs and pickleweed. Just over the bow of the kayak, you can see a small Spartina anglica partially covered by native plants found while kayaking in Fisherman Bay on Lopez Island, Washington. We had just reached out with the paddle to expose more of the plant before taking this picture. In the foreground, a Spartina anglica clone found while kayaking a log-filled estuary in the central Puget Sound. A single Spartina densiflora plant found in Grays Harbor about to be removed and transported to a landfill on an airboat. And as a quick aside, that boat and a crew are working southern Grays Harbor as we give this presentation. Here, a Spartina anglica plant found in the San Juan Islands is being dug and transported to a landfill on a small aluminum outboard-driven vessel. The kayaks are used to access shallow areas otherwise inaccessible to the larger boat. Earth Corps crew and Snohomish County staff being briefed on equipment safety before loading up on a Marshmaster and heading into the South Skagit Bay estuary for a day of Spartina survey and treatment. A Snohomish County employee collects Spartina anglica location data while a WSDA employee prepares to treat the plants. The next few slides will focus on Spartina distribution and eradication status. This site depicts the distribution of Washington's 202 Spartina infestations. On this map, the black triangles represent the 76 Spartina infestations, all species combined, that have met our eradication criteria. Here, zoomed into Southwest Washington, you can see the blue triangles along the coast, representing the 37 Spartina alterniflora sites still infested. These populations consist of a few hundred individual plants intermixed with tens of thousands of acres of native species. Also on this slide are 3 orange triangles in Grays Harbor, which represent the remaining Spartina densiflora sites. These 3 populations are comprised of a few dozen cryptic plants intermixed in over 4,000 acres of native species. On this slide, the 2 yellow triangles in Hood Canal represent our 2 Spartina patent sites, comprised of a few individual clones. And the red triangles in the North Puget Sound represent the 84 Spartina anglica sites, which contained over 30,000 individual plants in 2025. This slide depicts our 2024 eradication status. You can see that since the adoption of our eradication criteria in 2011, the program has made steady progress, eradicating 38% of Washington state's known Spartina infestations. As of this time, we're still receiving 2025 data from the cooperators. By February, we typically have all data compiled and finish our data review. Now, I want to talk about the 3 keys to our eradication success. First, a knowledgeable and dedicated staff, able to accomplish difficult tasks daily and impossible tasks with a few months preparation. Here you can see the list of the project partners that have made Spartina eradication in Washington possible. In 2025, we had over 80 field staff working on the program. The contributions made by every person are greatly appreciated. Second, viable eradication tools and techniques. Acquiring the equipment seen on previous slides, combined with our dedicated crews and a safe, effective aquatic herbicide, have been key to program success. That herbicide, an aquatic imazapyr formulation, first became available under the trade name Habitat in 2003. The third key to eradication success has been the political will and energy to carry out the program. This includes support, funding, and authority. Here's a snip of RCW 1726, in which the legislature directed and authorized WSDA to lead Washington's Spartina eradication effort. In addition, state and federal legislation has funded the program since 1995. So, we've seen the impacts that Spartina can have and discussed the success of the eradication program has had over the years. We just talked about the 3 keys to that success. Now I want to talk about the biggest emerging challenge, North Puget Sound salmon restoration sites. Specifically, the restoration efforts underway in Washington State's Water Resource Inventory Area 5, or RIA 5. You can think of this as the Stillaguamish River watershed, its delta, and immediate surroundings. These restoration efforts are reclaiming estuaries previously diked and modified for farming purposes. The sites undergo extensive and costly preparations before dike removal returns them to the estuary, where they play a crucial role in the successful outmigration of juvenile salmon. RIA 5, shown here, includes restoration sites critically important to threatened Chinook from the Stillaguamish, Skagit, and Snohomish River systems. Unfortunately, RIA 5 also contained over 16,000 Spartina anglica plants in 2025, or over half of Washington's Spartina infestation. RIA 5 salmon restorations include the Nature Conservancy Port Susan Bay restoration at 149 acres, the WDFW North and South Lekwe restoration sites at 288 acres, the Stillaguamish tribe of Indians, Zissabah 1 and 2 restoration sites at 291 acres. As of today, over 700 acres of restored salmon habitat has been added, increasing the area that must be defended from Spartina invasion by over a third. In the next few years, the Stillaguamish tribe of Indians has the Zissabah 3 restoration coming online, expected to add over 200 acres. WDFW's Johnson's Dyke restoration is expected to add around 15 acres, and other restoration sites are under consideration, one of which would add 90 additional acres. Once completed, the current and planned restoration sites will add over 1,000 acres of estuary for threatened salmon, simultaneously increasing the ideal Spartina habitat in Raya 5 by 50%. The story of Spartina threatening expensive restoration sites is unfolding as each new site is restored. The project partners genuinely want to defend this important habitat, but it is proving to be a challenge. Here you can see the Stillaguamish tribe of Indians Zissabah 1 restoration site. In 2019 and 20, after breaching the dike, a few plants were found. Almost 100 in 2021, close to 200 in 2022, 370 in 2023, and 570 in 2024. This increase happened even as our tribal, county, and state cooperators labored to find and eradicate this harmful invader. At the WDFW South Leckway restoration, the Spartina infestation increased over the course of 6 years. Fewer than 100 plants were present in 2020. By 2023, over 5,000, and over 12,000 plants were found this year. As with the Zissabah site, this increase has occurred even as WDFW staff have worked to eradicate every plant that they can locate. Without their efforts, this site would be well on its way to becoming a solid Spartina meadow. We are seeking solutions for this expansion of the North Puget Sound Spartina anglica infestation into these expensive restoration sites. Some of those potential solutions are increased funding and support to have larger crews able to spend more time looking for and treating these plants, shifting herbicide rates, tank mixes, treatment timing, or moving from spot spraying to broadcast treatments in the most heavily infested areas, and evaluating the possibility of herbicide resistance or tolerance. Sites like these have made the threat of Spartina damaging valuable salmon restoration sites evident. Moving forward, the threat of invasive species should be part of the planning and funding process as new restoration projects are implemented. Recently, WDFW and the Stillaguamish tribe have been communicating before rolling out new restoration projects, and their staff have been seeking funding to protect their projects. Ideally, securing funding to maintain these sites will start to be a part of the planning process from start to finish. The project partners remain dedicated to protecting Washington's valuable natural areas, estuaries, and restored sites, safeguarding the ecosystem services needed to support viable habitat for salmon, the food web, and future generations. We're reaching the Q&A portion here. I'd like to just quickly start out by answering the question I would have if I had just come into this, and that is, what is Zissabah? Zissabah was a prominent Stillaguamish tribal leader from the area before the dikes were installed in the 1800s, altering the landscape and harming the Stillaguamish River salmon runs. Eva?
Great, thank you so much, Chad. So a reminder again, you can use the Q&A box to add in any questions you might have for Chad. I can get us started. I wonder if you could just delve a little bit more into what exactly about those salmon restoration sites makes them so vulnerable to Spartina anglica?
I can. Most of these salmon restoration sites that are being installed right now are very ideal habitat. The way they're graded, the way the intertidal ditches are put in, how they function to be ideal for juvenile salmon to be able to use them. The way they want the water to flow slowly inside of these areas. It's really proving to be a perfect way to catch Spartina seed. And then the elevation, intertidal elevation at which these areas are being built is ideal for Spartina to infest.
Great, thank you. And we have a question in the chat about what advice do you have for Oregon? And presumably from Oregon, is saying we have currently low levels of Spartina densiflora in a few estuaries. All other Spartina infestations are presumed eradicated.
I would be very interested in having somebody reach out and contact me about that. We can do some more, just have some discussion on it. As far as Spartina densiflora, we have found that it tends to be tolerant to the herbicides we have available. So we can reduce populations with spraying. But once we have reduced them down as far as herbicide can do the job for, then we have to switch out and do the hard and difficult job of digging and physically removing them and taking them to a landfill.
Thanks. And I'll just follow up on that. It sounds like then, has there been any coordination, like particularly for the Columbia River Basin, on interstate Spartina efforts?
There has been, in fact, the West Coast Governors Alliance on Ocean Health used to sponsor annual interagency discussions that also included British Columbia. That is went away over the years, but I haven't lost track of everybody who was part of it, and we do still communicate.
Great, thank you. Move on to another question. Do you have a rough estimate of how much has been spent towards eradication mitigation of Spartina to date? And maybe I'll add on to that of just kind of it sounds like there's a lot of effort and presumably a lot of cost that goes into this effort. And I wonder if you could talk to maybe the longevity of this.
Well, the program started in 1995. And over the course of the program, it's somewhere in the $40 million between all federal and state, county inputs. And right now, the program is around $1 million a year. Those are all very rough figures. Did I answer the question?
Yes, I think so. I'll maybe follow up on that of just, do you think at a smaller scale, is eradication possible on a smaller budget?
Well, I definitely think it's possible. As the one slide showed, we're approaching 40% of all the sites that have ever been found in the state being eradicated. The bulk of those were smaller infestations, and the larger infestations are proving to be a little more challenging. But I think it's a very attainable goal to eradicate Spartina in Washington state.
It's pretty amazing. We'll have another question on if you've published any studies or maybe just reports on how Spartina, different species of Spartina move within a watershed, or are there models to show and predict that movement?
There has been a lot of work done over the years. There were some pretty major drift card studies done years ago looking at the likelihood of seed dispersal, both on the Willapa and the Puget Sound infestations. If somebody reaches out to me, I'm sure I can try and help them find those studies.
Great. Thank you so much. Looks like we're having still a couple more questions come in. I am going to take a moment just to say that if you're still on here, I am going to make an exit poll available in just a moment. So do definitely stick around for that, please. And I'll also ask for our panelists to maybe just put their contact information and any resources that they'd like to share with the audience in the chat box. Panelists, you should be able to toggle that to go to everyone. But while we're doing that, Chad, we have a couple more questions. Maybe I'll start with, is the application of habitat, the herbicide, being studied for its effects on species to make sure it's not having adverse effects?
I don't believe there's much work going on currently. There were vast amounts of work conducted before it was allowed to go out into the marshes as a commercially available herbicide. And quite a few studies have been done over the years. Again, people can reach out to me. They can find me if they use the QR code there or just call the 1-800 line. But we're very confident in the way we're treating, that we are not harming native species. In fact, we are creating a space for those native species to come back in.
Great, thank you. And then there's the question of if, and maybe I'll also follow up of to what extent has there been just like hand mechanical removal and excavation of plants? And in those cases, is there a need for a cultural resource survey first?
Is there a need for what?
Cultural resource survey?
We definitely work with the tribes. How we deal with mechanical removal varies by site. I was just last year, I was out with the Suquamish archaeological staff, and we went through one of the sites and looked for anything that could be of concern. We definitely work with them. We worry about impacts to cultural items that could be out there. I suggest that the crews keep an eye out. This is one place where I'm actually more comfortable with using herbicide than digging for removal. Though when it comes to something like I was describing, densiflora, the only way to get the last of the plants is usually to dig them out. So we just try and be very aware of what we're digging in, and if we see anything, and we would notify anybody if we saw something suspicious.
Great. Thank you so much, Chad. All your exit polls should be live now if we'd like to take a few moments to go ahead and answer that. We have some resources in the chat now. I'll put a couple more announcements in there as well.
It looks like there's one more question that just came up in the chat.
Thanks, Darcy. Yeah, Chad, that's for you. Has this been found in the Snohomish River? So Spartina been found in the Snohomish River restoration areas like Smith Island?
It is getting close. It's on Tulalip tribal ground in the area of Big Flats. So would that be north and west? But I don't think it's made it into the new Smith Island area. I think that might be a little too fresh water. Though we've definitely found some knotweed up in that direction.
Great. Thank you so much. Thank you everyone for attending, and we'll send an email with the recording of this and those links to the resources again, just in case. Thank you so much, Chad and Michele, for your time. Those are great talks, and it's really nice to see how these two things fit together from these different perspectives as well. Thank you all and have a great day.
Thanks, Eva.
Thank you, everybody.
Thanks, everyone.